Great. So next up, we have more of a talk rather than a pitch. We have Stef van Grijken, who is the co-founder and CEO of Cradle. Now, he's going to give his perspective on the intersection of AI and biology. So this is really checking in on Googler startups and how they evolve. And today we're just seeing a lot of them at the start of the journey, but this one's more, what happens next? So, hi, Stef. Have you got slides as well? You're going to post them up?
I do. I think they're going to come up right now. Thank you for having me. Big fan of the XFi community, and we thought, as fellow Googlers building a business, it might be helpful to build a bit of a community and talk a little bit about maybe a less obvious domain, which is applying large language models to engineering biology, which is really what we're trying to do at Cradle. So we're a team that got started in 2021. A couple of folks, Eli, who was part of Google Brain before, Daniel, who was one of the early folks in the Zurich office for Google. And then obviously, as we're in biotech, so having biologists is quite important as well. So what do? We love to make it a bit easier to program biological systems. Now, obviously, biology is incredibly complicated. And so at the level of cells or even whole organisms, we don't think there's a lot of help yet, unfortunately, for generative models. That said, some of you might be familiar with some of the work that AlphaFold has done in sort of rendering and showing what these molecules are like. What we're trying to do is really, I want to improve this molecule in a certain way, and sort of what will be the sequence that I need in order to do this. Biology is already quite central to a lot of our lives, whether that's in medicine or chemicals or materials. A lot of proteins are being used in those products. According to McKinsey, almost 60% of all of the products that we consume today could be made with biology. And just to give you a sense of some of the products that are out there, about 60% of all of our pharmaceutical products are based on proteins. So whether that's antibody therapeutics to vaccines to many of the diagnostics that you might have been familiar with even during COVID. But maybe a little bit less known, you might have wondered at some point, why do my detergents keep working at lower temperatures? That's basically because people have been starting to mix in enzymes that are much better at breaking down fats and starches and those types of things, all the way from degrading plastics to nutrients to serving the biotech industry today. So these are incredibly powerful little machines, but they're very hard to engineer with some of the tools that people are using today. And it was interesting, the previous talk actually made me think of how a lot of folks in biology use these very large physics simulators as well, and might be waiting for weeks to months for them to figure out how a protein might fold or might interact with a certain other molecule. So the sad reality of being a team within bio is that you go through several dozens, sometimes even 100 set of experimental rounds where you're trying to improve your enzyme product to make it do something that nature didn't intend at the beginning. Every experiment, so every label costs about $150, and then with the methods that people apply today, maybe 1 to 5% of the candidates where they've changed the sequence come out being improved compared to the ones that they had before. And so I think one of my favorite quotes from one of our customers is sort of 99% of our experiments fail in the lab. We just don't know which 99%. We do know it's about 90% of our runway until Series C. And so that makes building a bio-based product a very expensive thing to do. So we use language models in a generative fashion, but instead of training them on text or code, we trained them on protein sequences as well as experimental outcomes that we've generated within our own lab, such that you can use them to improve upon existing proteins that you might be working with. So for example, I would love my protein to be more stable, which means that it can endure higher temperatures, or that it's more active, which means that it can do a certain chemical reaction faster, or bind better if you're in a therapeutics context, sort of attached to a certain molecule that you wanted to attach to for a longer period of time. We can do that across some of the properties that a lot of these biologists care about. So what you see here is a little Y-shaped protein called an antibody. The top left side might want to attach to a cancer or something your own immune system doesn't recognize as a threat. And you can basically ask our models, hey, I would like to bind better to this thing, be more stable, and look more natural to the immune system. And we will effectively generate a set of sequences that will go into your experiments as a R&D team within biology to then sort of serve its purpose. Thank you, Natasha. I am not an MD. I'm one of the more mathly inclined people. We can solve for several types of tasks today, especially within, let's say, the realm of planetary health, where people use a lot of enzymes. So stabilizing those molecules and making them more active is something that's completely self-service in our software today. We're currently piloting expression, which is making more of the protein within a cell: affinity, specificity, cell penetration, and then stability. Think of these as sort of, the Figma tools that a lot of biologists would like to have. This is a SaaS, and so our customers will pay for every molecule, like every protein that they're engineering, and they use it as part of their sort of the design step in their design, build, test, learn cycle. Within our lab, folks are typically research associates or scientists that use our software, sometimes even medical doctors. Like I said, we license out access to the software through the web, which honestly in biology is already quite fascinating, because a lot of things are happening still on people's local machines. And then over time, we also love to sort of allow people to run certain experiments remotely that they're doing sort of physically in their own lab right now, and really with the goal to just dramatically reduce the amount of cost and time that you need in order to bring a bio-based product to market. Just to give you an example, we have our own laboratory. We do three things there. One is it's generating labels. So we try to test out whether our models generate the right type of stuff. Secondly, we built these demonstrator projects, which is to show to other folks in the industry that our models actually work. And then thirdly, good Google tradition, we eat our own dog food. So our own research associates are using our software in our lab, and we try to make it better that way. This molecule is called T7 RNA polymerase. It's the molecule that turns DNA into RNA. You can sort of see it in the middle with the helix being unraveled there. A lot of people that are making RNA-based drugs, this will be their manufacturing workhorse. So this is the enzyme that you use in order to produce your RNA-based drug. If you can make it faster and more stable, you can effectively keep the reaction going longer, and then you can produce more product for the same amount of cost. And so that's something that they typically like. Plus, you can make longer products, so you can have, quote unquote, a more complicated RNA instruction being manufactured. So typically, our customers get between 1% to 5% hits when they sort of make changes to this molecule. For the first set of ones that we generated, we had around 70% of our candidates improving both stability and activity, and introducing a lot of mutations that nature hadn't come up with before. Now, given this thing polymerizes RNA, it's like highly conserved in evolution. Like every organism has this. If you break this enzyme, like nothing happens anymore. And then what you can do in the subsequent round, which is actually pretty cool, is you can sort of fine tune your models and see, hey, could we generate candidates that sort of are very predictive of what might happen in your laboratory? So what you see here, the blue dots are generated sequences with the model's expected value for what was going to happen in the actual experiment in the lab. And then the red dots is what actually happened within the laboratory. So we found 95% hits here, and the correlation between our expected lab outcomes and the real results in the lab were a 0.83 correlation when you use Spearman rank coefficient as the measure of success. We have 9 customers right now. We have closed our Series A, and we're currently scaling to work with a lot more customers. Our current customers include a top 5 global pharma, top 5 global chemical player, and a top 5 global biotechnology player. And so we're quite excited to grow the business right now. But unfortunately, not looking for investment at this point.
Amazing. I can't believe how much you've done. It's such an interesting topic and a crazy area to be working in. You say you're not interested in money at the moment. What's the kind of runway looking like? What are you going to be doing? What's the future hold?
Yeah, that's a great question. I'm not going to comment on the exact size of the runway, but plenty at this point. I think for us, it's really about, like, most of our work was in can we build models that are actually dramatically better than what humans can do today, which I think we've been successful at least for enzymes. We're now scaling into peptides, vaccines, and antibodies as sort of new modalities that we can work on. We're also, growing our commercial operation and getting to a point where, let's say the platform right now is lots of duct tape and tie wraps. And so we're trying to make that into sort of real software, or at least I want to be able to sit in demos and not be embarrassed as a former Google PM.
Absolutely. Another former Googler has asked, who are your existing investors? That's Jürgen Galler.
Thank you. Yeah, we have Index Ventures and Kindred Capital as our current investors.
Fantastic. Now, Alex Belenke has asked, what are you so interested in? Is that your question?
Sorry, I didn't hear a question in that question.
I know. Alex, do you want to come online and ask that, or reiterate it? I don't know whether I think that was.
Hi.
Oh, there we go.
Yeah,
Hi.
I was wondering if you're not interested in money, but you anyway decided to join the call. Is there anything that you may ask the Googlers community or Zooglers community for?
Are you hiring?
We're hiring, certainly. So definitely, if you're looking for a job, check out jobs.cradle.bio. Yeah, I mean, a bunch of the XY team folks, we were chatting, and they're like, "Hey, it would be super fun to just showcase some of the work that some Googlers are doing in Zurich." So I didn't come into this meeting with a particular goal in mind, but anyone that's interested in this stuff, research wise, or if you want to talk about other types of collaborations, happy to.
And we will definitely facilitate any connections, networks. If you want to be put in touch with people, we will do that. So yeah, fantastic. Really interesting. Thank you, Steph.